we realize we have microbes in our gut and we have microbes everywhere, and it fulfills all kinds of functions. So leaf microbes are very essential for these oak seedlings, and also in the roots it has all kinds of microbes that either try to kill it, or it helps it to take up water, or it helps it to resist different diseases. So that's kind of the study organism in this talk. But so what may happen if the climate changes, or we have spatial variation in climate? What may happen with this oak seedling and everything that's associated with it? So this is some answer. This is a paper from Thackeray in Nature a few years ago, and they made some predictions for spring phenology. And there on the left you can see that they predicted that primary producers, like oak trees, they might advance their spring phenology, like when their leaves appear maybe, by just 2 days. Primary consumers, like insect herbivores, plant pathogens, some endophytes, they were predicted to advance by 5 days. They didn't look at microbes actually, I think. And then you have the secondary consumers, so the ones that eat the primary consumers, like parasitoids. And they were expected to advance 2 days. So some of the questions we tried to address in my lab is what are actually the patterns we expect in autumn? We know what happens in spring, like that will advance spring. But what will happen in autumn? Phenology is just the timing of things, when things happen in nature, like when buds burst and when leaves drop. And also another question, how will species interactions change with climate? So this talk, I want to talk about the work of 3 persons, and I put it into 2 questions. So the first one, what is the impact of a changing climate and genetic variation on plant phenology and its associated pathogens and microbes? So what's the effect of climate there? We did a large field heating experiment, and every time I say we, it's actually someone else who did the work. But every time it's not always the same other person who did the work, if you get me. So this is the work of Maria Fatikov. Then we did a small controlled growth chamber experiment to look in more detail into the microbes, and that's the work of Ahmed Abdel Fattah. And after looking at the effect of climate on phenology and microbes, we wanted to understand also what's the impact on insects, and in particular insects that may be present with multiple generations during the year. And this is the work of Alvaro. So this is actually all have received either some pilot funding, or Alvaro is doing his PhD as funded by RA8. So Maria did a field heating experiment, and for this we basically established 2 treatments, 2 cages with 2 different temperatures. In each cage we had oak trees of 5 different genotypes. And then during the growing season we followed bud burst, leaf autumn phenology like leaf coloration and leaf drop, and also the natural attackers like powdery mildew pathogens. I'll use this modern pointer here now. And also we took samples for the endophytes. That's what she's currently working with. So here is how it looks like in real life. So you see the different cages, and on the top right you can see the ceramic heaters that we use to heat the cages. So what happened with spring phenology? So this is on the y-axis, you see some kind of scale reflecting the level of bud burst. And what you can see is that if you look at the graph, you see the response here of 5 different genotypes. Those are the different colored lines. So for each of the genotypes, the warming accelerated the tree phenology. The 3 genotypes, the different lines, they differ in their spring phenology. And the 3 genotypes respond somewhat differently to warming. So okay, spring phenology advances with a warming climate. So what happens in autumn? So here you see the response again, the phenology on the y-axis. And you see that with warming, the leaves stay green longer. So leaf coloration is delayed with warming, But this response varies among tree genotypes. So you see that some of the lines are horizontal. They don't respond to warming. Here's a graph for, well, actually not a graph, but just photos for leaf drop. It's quite striking. On the top, you see the heated cages, and the photo taken of the non-heated cage. Below, you see everything has dropped their leaf, and above it still has the leaves. So there's quite a strong effect of warming on leaf drop. So here's the effect on one of the main pathogens on oak trees. It's a powdery mildew pathogen. It's the white fluffy stuff on top of oak leaves in the late autumn. And you see that warming actually reduces the level of the pathogen in this case for all of the 5 different oak genotypes. So mildew decreases, and this is fairly consistent across the different genotypes. So in this case, a disease is not getting worse with climate change, which is not a thing you hear every day. So now within the same experiment, we're actually looking at the leaf microbiome. So how does the warming and how does genotype affect the microbes? So we don't have the answer yet. We just got the sequencing data. And the only thing we looked at actually, we took leaf samples across the growing season in spring, summer, and autumn. And this is the very first and only plot of the data. And here you see a multi-dimensional space, and you see that the leaves in spring have a totally different fungal communities than the leaves in summer and autumn. Yes, so this is all work in progress, right? So this is all we know so far. Next time I'll tell what are the other answers. So there's very strong variation in the leaf microbiome throughout the season, which I think is quite interesting per se. But where do all these microbes then come from, and why do they change throughout the season, right? And how do the first microbes get into the leaves? So for that we did actually, and in this case I mean Ahmed, he really likes the kind of small scale controlled experiments. So we grow the acorns in small tubes and small microcosms. And then we actually look, if you just put an acorn in a sterile environment, how do some microbes go from the acorn to the leaves? And where do the microbes in the roots come from? So that's the question he asked. Where do the microbes in the leaves and roots come from? Do they come from the acorn? So he grew it in a sterile environment. And the answer is not very clear on the slide. I was actually just over lunch asking him the answer because we got the sequencing data. So most of the leaf microbes, they actually come from the embryo. So from the green part inside of the acorn. Most of the root microbes, they either come also from inside of the acorn or they come from this hard shell that surrounds the acorn. So many root microbes, they're hiding in the acorn shell. So he's now following up with looking how this microbial community is affected by climate. So what's the impact of climate and drought on the leaf microbes, and what about the microbes in the roots? So he just started an experiment, 3 different climate treatments, 2 different soil moisture treatments. He likes kind of fancy setups, so he built his own climate-controlled cages, so he can follow live on his phone, like what's the humidity in his different pots and cages. So here you see the different boxes he's been building. So this is a photo from yesterday, actually, because you want to replicate boxes, right? You can't do it in just one growth chamber. And if you open the boxes, you see the acorns and the separation between the soil and the air, and you see the soil moisture meters and the temperature sensors. So as I just told, this is how it looked yesterday. So the plants don't have leaves, and the answer is not yet there. But the question again is like, how does temperature and humidity, how does it affect how the microbes and which microbes will move to the leaves or to the roots? So let's go to the next question. So now we know like climate may affect the plant phenology. It may also affect the microbes that associate with it. And do the insects that eat the plants, do they care about that? And why am I interested in multi-generational insects? Well, I just mentioned that the climate may affect the phenology of oak trees, right? Here you see that through time, the oaks start to burst their leaves earlier in some small place in the UK. With insects, also another phenomenon may happen. So here, if you look at the dashed line, you see that this particular insect, the four-spotted something moth, it was active really during this period of June, July, and August, before the 80s. So the dashed line is before the 80s. But after the 80s, it became active in this period, and then once more. And the reason is due to climate, rather than having one generation, it could suddenly have two generations during the year. And this is what Alvaro is interested in. Like if you have insects in Southern Europe and in Northern Europe, and it's the same species and it attacks the same plant, may it change the number of generations? But I didn't want to talk about this geographical study, but I was interested in more answering the question, what's the effect of leaf phenology and pathogen infection on insect attackers? So Alvaro looked at different attackers, the free-feeding insects, some leaf miners, and aphids. So he created 6 treatments in spring. So on the x-axis you see the phenology treatments. So some seedlings he planted very early, some in the middle and some late. And on the y-axis you see that he infected some of the seedlings. And he kept some other seedlings healthy. And then we tested the effect of these treatments on the first, second, and third herbivore generation. So do herbivores prefer or perform differently on different kind of plants? And does this differ between their different generations? And then when I prepared to talk, I was realizing this is getting very complex to explain. So Alvaro sent me this graph, and I was like, I'll never manage to really talk through that whole graph in this presentation. So I thought I'll just focus on one very small subset question. Let's focus on the aphids. So what does an aphid prefer, and does its preference change through the season? You can also interpret that like, is it selection pressure on different plants of different phenology different throughout the season? And what may be the effect of having one more generation of aphids if the climate changes? So there are a lot of questions a little bit intermixed here. So when we look at the different treatments in spring, the answer is as follows. So in spring, aphids prefer actually the late phenology plants. If we move to summer, the aphids prefer the healthy control plants. So in summer, they don't like the infection by the disease. In autumn, they prefer the plants with the middle phenology, and they care less about the infection. What this all means, I'm not sure yet about. We're kind of trying to interpret, but I think it's very interesting that you may have different selection pressures, and the advantage of trees to emerge early or late during the spring that might change when the insects change their number of generations. So here's some concluding thoughts. It's work in progress, of course, but warming affects both spring and autumn phenology. The warming did clearly affect the specialist pathogen. The pathogen did less well with warming in this case. And currently we look whether warming affects the leaf and the root associated microbes. And the last point is very vague because we're not there yet. Like climate change may change the interactions between plants, microbes, and insects. That's kind of the topic we're pursuing. So just once more, the persons who actually did the work, and thanks for listening.
Okay, we have time for questions. And for those of you that arrived late, that you need to speak into the microphone so that the people listening to the streaming or in the room besides can hear you. So.
Okay, so the oak genotypes that varied in the response, where did they come from? Were they from the same population? Were they from everywhere? Or I mean, what is the origin of that? And is it sort of, I mean, that's interesting in the sense of it shows that there is some raw material for evolution to work on, and what is going to happen in more in natural populations.
Yeah, we just got the reviewer comments, and they asked, no, you say it's interesting, but that's, no, that wasn't that bad, but yeah, one reviewer thought you couldn't say anything if you had only 5 trees, but luckily the subject editor disagreed a bit, but they were interested in the origin. Yeah, they come all from the same island. So it's a 5 square kilometer island, and they come from there. So I think it really means there is genetic variation within what I'd call a population, but otherwise it's at the landscape scale. Yeah, I think that makes it very interesting. Yeah.
Okay, thank you. I think we're running out of time. We could talk to Aiku during the break, and then we'll have the next speaker.
Yeah, Now I know where all of a sudden this tree showed up, because I was standing here and 10 minutes later all of a sudden there was a tree. I think that was fast growing. So I didn't introduce myself before. I'm Frederick Schenk. I'm one of the research area leaders of number 5. So that's basically climate on centennial to millennium scale. Another research area leader is Malin Schillander. She's also sitting here, and her 2 PhD students will give the next presentation. And first it's Eleanor. So ready with the hairs?
Okay. So maybe. So. Okay. Okay, I'm a PhD student at the Department of Geological Sciences, and I'm a member of the Bolin Center research areas 5 and 8. And my project is about spatial and temporal changes in species distributions in boreal peatlands, where I study peat from south-central Sweden by using macrofossil analysis. So in this presentation, I want to introduce my project a little bit and share some preliminary results from and next steps for my first site. My project is essentially a combination of paleoecology, geoscience, and paleoclimate research. I work within 3 boreal peatlands in south-central Sweden. Oh, I need to have the pointer. So we have Stora Mosse in the center, and Draftinge Mosse to the left, and Dala Mosse to the right. I look at species, changes to species distributions and vegetation transitions with a specific focus on sphagnum mosses. And this is because we want to understand how species within these peatlands have responded to change in the past. My aims are therefore to assess and identify internal successional processes and biotic interactions, as well as the external climate drivers of change on spatial and temporal scales. My sites are within an area that's been previously researched in our group over the past 10 years, but also researched by other people for about 100 years. It's in an area that was once covered by a glacial ice lake, which is modeled in this map within the white lines. This lake eventually drained and allowed for peat to start accumulating. This means that my sites are essentially in the same system in terms of the regional climate, geomorphology, and the ages. In this area, the peatland development, or the normal peatland development, is usually from a lake into a fen and then into a bog. But in my site, we found something slightly unusual, which I will talk about a bit later We're interested in boreal peatlands because they play an important role in the biogeochemical cycle of carbon, where northern peatlands store approximately 1/3 of the global soil carbon. This is of importance to investigate these changes within these terrestrial ecosystems, because we want to see how this may affect carbon storage and release in the future. The dominant species in these peatlands are sphagnum mosses. They tolerate highly acidic and nutrition-poor environments and tend to preserve well thanks to their decomposition-resistant qualities. I look at these mosses, among other vegetation, using macrofossil analysis, which is a method where you look at plant remains to see what they can tell you about the climate, or dead plants as well, to see what they can tell you about the climate and environmental conditions from the past. So peat is almost entirely made up of plant remains of the vegetation that once was at the surface of the bog or peatland. Is therefore very suitable to macrofossil analysis and also a quite local paleoarchive. But before I can do any analysis, some field work is required. This video is from a year ago when we were coring in Dalamosse. So we core 3 sequences per site, A, B, and C, and 1 sequence per site will be the high-resolution core called the master core. All the material is subsampled before analysis at 1 centimeter resolution in preparation for a range of methods, including AMS radiocarbon dating for our age model using the software R and the Bacon package. Then a macrofossil analysis for species identification and identification of vegetation transitions. Bulk density alignment for core, analysis for core alignment, and this is within each sequence but also within each site. Then we use the age model and bulk density to calculate the peat accumulation rates throughout the sequence. I will also, at a later stage, look at the species distribution modeling in my sites. So for my first site, Stora Mosse, sequence A is a more higher resolution core, the master core, and this is a picture of a section of the sequence at about 4 meters depth, where you see quite well preserved sphagnum. So I'm just going to get into our age model. We see the depth on the left and the ages down the bottom. This model is based on 10 age dates that from macrofossils that I picked, and you can see them represented in blue. The grey areas are the uncertainty that the model has put in. We have recently received 6 more dates just the other day, so we haven't had time to put it in the model yet, but it's very exciting still. This sequence is about 7 meters in length, so our oldest sample, as you can see, is almost 10,000 years old. In the macrofossil analysis, I've analyzed 72 samples so far throughout the sequence, and I've focused on 4 main areas. This is falling off. Okay, sphagnum in green. Okay, sedges in orange, and then dwarf shrubs, other bryophytes, and unidentified organic matter in grey. Okay. I've also noted or counted charcoal wherever I find it in the sequence. Thank you. So if we have a closer look at the first part of this diagram, we have the depth again on the left and simplified peat stratigraphy. The different things I've found in the sequence at the top, and it's either a coverage percentage or a count. Starting from the bottom, with the lake draining, turning into a fen, we see an increase here in the typical fen vegetation, sedges, and as well as a more highly decomposed material. This continues for 1.5 meters approximately, so at 4.5 meters into the sequence we see a change, and this is where the system transitions into a bog. There's a lot less sedges and a lot less decomposed material. But something interesting happens at a later stage at the, or 2.5 meters in and 1.5 meters into the sequence, where the sedges and the decomposed material come back again. And as you can see, this also aligns quite well with the charcoal phases that I found up here. If we look at the sphagnum part of the diagram, this is associated with the bog stage mainly. What we can see in the bottom right, in the fen stage of this system, we have a few occurrences of the species that are more associated with the fen, but then at 4.5 meters depth, we see a significant increase in the more typical bog sphagnum species for boreal peatlands, and this is Sphagnum fuscum and Rubellum. This is mainly hummock species, a little bit drier, and changes gradually into sphagnum medium, has a slightly broader specific conditions, and then it turns into the wetter hollow species of Cuspidotum and Maius. If we look at all this and put it together with the bulk density in blue and peat accumulation rates in grey, with the ages on the left-hand side and the simplified peat stratigraphy again on the right, we start to see some interesting stages in the peatland. Starting with the lake again, where the system changes around 9,000 years ago into a minotrophic fen. This is where the system receives both atmospheric input and groundwater input. You see that it also has a slightly higher bulk density. At about 6,300 years ago, the transition into an ombrotrophic bog happens, the fen bog transition. This is where the system only receives atmospheric input, and you can see a lower bulk density as well, and it's mainly sphagnum in this section. And then we have two interesting events. Around 4,200 and 2,200 years ago, we see the fen-like vegetation coming back again with slightly higher bulk density and almost no sphagnum at all, which is something that we're currently investigating. Another thing to note is that we have periods of higher peat accumulation rates. This one around 5,000 years ago has been previously identified by my supervisor, Bolin Days, in the same peatland as the high peat accumulation event. It's also been identified by my colleague Jenny Sjöström in Draftingemosse, which is a site that we share, and she will talk more about this after my presentation. But we can also see towards the top of the sequence some interesting peaks in the peat accumulation rates. And finally, we see the two charcoal phases again lining up quite nicely with these two unusual stages or transitions that we see. So far we've looked at the successional processes and found some unusual things in our peatland, but we can't say much about the biotic interactions yet. The next steps are to correlate sequences A, B, and C within Stora Mosse and have a closer look at the species distributions, and then do the same thing for my other two sites, Dalamosse and Draftingemosse, and eventually compare and contrast across all three sites, and also link these with local climate records. So in conclusion, the preliminary results for my first site, the age model, bulk density, and peat accumulation rates align quite well with previous research. However, the macrofossil analysis has revealed some unusual stages. And the next steps are to look at the species distribution and then compare across the sites. Thank you for listening, and thanks to Bolin Center for funding my project and the grants I received. I also have a poster later on if you're interested to see the details. Thank you.
Yeah, thank you very much. And I think it was your first public talk, and I think that was very good. I mean, I think I wasn't that good at my first talk. So,
well, thank you.
Yeah. Any questions?
Yes, thanks a lot for a very nice and interesting talk. One of my issues is that we try to see climate as both temperature and of course moisture. And this particular peatland, it is to a large extent regulated by the groundwater level and of course the height of the big bog. So I wonder, your growth rates that you have seen on the peat, if you have tried to correlate those also with the hydrological issues. Since you know a peat big bog will collapse later on when they cannot hold all the water, because this is also on the position of the height of the bog. So those connection to the hydrology, I'm curious about.
Yeah, I haven't yet looked at this because haven't got to that stage yet, I guess. Yeah, that's I can't answer that question right now, but I will hopefully in the future. Yeah, that sounds great.
Yeah, I think one reason why she does that research is actually to find replies to that question. So I think, yeah. Do we have time for another question? One? Okay.
A great talk, by the way. And you described those two little bursts of fen-like vegetation that were sort of, I gather, in a bog-like state as being unusual and associated with some kind of burn. But why is that unusual? So I'm not too familiar with the literature. Is that not it's not a common pattern that you see? To have sort of a flip-flopping between states?
It's not very common, and I don't think it's been seen in this area before. But we don't really know why it happens and like how big this change is. But that's what's going to be interesting to see in the other two peatlands, because they're a lot smaller. And if we see a similar thing happening there, we can see how fast and how slow things are happening. And yeah.
Thank you very much. I think we have to move on to Jenny. She actually, more applause. So Jenny shares some of the sites of Eleanor, which we also like as a research area because it makes our funding situation much more efficient. But they actually study partly quite different questions, so it's very interesting. That's why we also put them just next to each other to see how many questions you can actually maybe try to study based on these peatlands. Yeah, and I made a joke with the hairs before, but it's not really a joke. I know that from my personal experience. So it's actually quite unfair for people with long hairs. So, but you managed. So, yeah, welcome.
My name is Jenny Sjöström, and I'm a PhD student at the Department of Geological Sciences, also here at Stockholm University. And as Eleanor mentioned, we are in the same group, but Eleanor works on the organic part and I work on the inorganic part of peat sequences. And we share one site, which I will present results from here. So I work with paleodust deposition, and I use raised peat bogs in south central Sweden as an archive for this. But first, I want to say something about why we want to study paleodust deposition. So atmospheric dust particles, here represented by the orange cloud, affect the climate system both by absorbing and scattering incoming solar radiation. This process also brings nutrients to nutrient-limited both marine and terrestrial ecosystems. And currently, the majority of paleo studies over longer timescales have been done on marine cores, even though the majority of dust is actually deposited on the continents. So lower latitudes are more studied in this context than higher. And the area that I work in, Scandinavia or Sweden, is only represented so far by one paleodust study, and that's been done on Store Mosse, which is the site that Eleanor was talking about, and my supervisor Malin published that study in 2013. My site, Draftinge Mosse, is located just 15 kilometers southwest from Store Mosse. It's located in a granitic bedrock setting, so we expect to find minerals such as quartz and feldspars, muscovites, and maybe some clays. And we find peat bogs particularly useful for for this kind of reconstructions because they receive atmospheric input and thereby act as an archive for atmospheric dust deposition. The aims within my PhD project is to compare and contrast paleo dust deposition within 3 sites. So I work with Drafting mosse, but also Hjälsred mosse, which will be the next step for me. And then I will compare the events from all 3 sites. So before I can do the analysis, we need to go out in the field, and usually in weather looking like this. And we do Russian coring, and we take out cores and that are later freeze-dried and cut into centimeter slices. Each slice is then weighted and traced in order to calculate the bulk density. Thereafter, some parts of the samples are used for radiocarbon dating so that we can get ages to the changes. And the rest is milled and undergo various geochemical analysis. And I've done analysis both on bulk samples, but also on ash samples, when we've removed all the organics. And I've also done, which is a bit unusual in peat paleo dust studies, but I've also done X-ray diffraction analysis. And this is a way to directly identify minerals. If you work with elemental data, then you infer the minerals from the elemental compositions, but the benefit with X-ray diffraction analysis is that you actually get the minerals identified based on the cell structure. And this gives me a quite large data set that I use statistical principal component analysis in order to see which elements behave in a similar way throughout the profile in order to identify governing processes. So now we turn to the results, and this is the HDAF model from Drafting mosse. It covers 8,300 years. 16 samples are used in the HDAF model, and it's done in the software Bacon. 2 dates are not used. You see the red stars, and that's based on bulk dates, and they showed consistently older ages than nearby samples that were based on plant macrofossils that were picked out. So they were not used in the model. Here I also want to make you aware of that we have a period with very low accumulation rates, and that means that for the coming slides we will have a lower temporal resolution around this time period. So these are some of the results from the peat property analysis, and I just want to first say that on the y-axis we have the ages. So it's we have the present on the top, and we go downwards in time to 8,300 years ago. And I won't go through all the details of the data here, but as I mentioned, when working with paleo dust deposition, it's important to know that the system is ombrotrophic, i.e. that we receive the minerals from the atmosphere exclusively. And then we look at a combination of the peat composition, the ash content, and some other concentration values to see when did this transition occur. And at Drafting mosse, this occurred around 5,500 years ago, the transition from a fen into a bog. And on this slide, I also want to make you aware of that we have a very rapid increase in the peat accumulation rates. We have several of those. I'll get back to that, but just want you to note that. As I mentioned before, I do principal component analysis of the elemental data. And this groups the elemental data sets into components that show a similar behavior throughout the sequence. The first component, there we find a lot of lithogenic elements, and that's elements that are associated with minerals. And this is the variation of this component through time. So it has a larger weight in the lowermost part, in the fen part, compared to the ombrotrophic part. The second component, there we find elements that are associated with soluble phases, or phases that are not stored in the system, such as calcium and iron that can be transported out with water. And in the third component, there we find elements that are both associated with lithogenic elements, but also with elements that are recycled at the surface. So we have lithogenic elements, or the elements associated with minerals, split into two components. And when trying to put this together, we can see, based on the behavior of the elements, have selected elements that are known to be of a lithogenic and have a conservative behavior in peat, and calculated mass accumulation rates for scandium here. And based on this, you can see that there are 5 periods of increased mineral input in Draftingemosse. The first period is the most significant. This is actually on log scale, so that event is really big. And from the X-ray diffraction analysis, only 2 phases could be identified, and that was quartz and an amorphous phase. In the later stages of increased mineral deposition, typically felsic minerals such as quartz, feldspars, muscovite, and also some clays are identified. And I also here want to make you aware of that we have one part of the sequence around 5,500 years ago where we have a lot of carbonates that were later found to be of autogenic origin. And the elemental data can also be used to infer grain size. Here I've plotted the silica against titanium in order because that can say something about the grain size. So in the lowermost part of the sequence, in general, coarser grains seem to have been deposited compared to the ombrotrophic part of the sequence. And this could mean either a different transport process, i.e. fluvial in the fen part of the system and aeolian in the upper part of the system, or it could also say something about wind strength. And as I mentioned, the aim is to compare my results with the previously conducted paleodust study, and I'm still working on this, but here I've plotted the peat accumulation rates of and scandium mass accumulation rates from Draftingemosse together with the results from Storemosse, which is the previously studied nearby system. Oh, this is falling. Sorry. What we can see here is that there are a lot of similarities. However, the magnitude and sometimes the timing of events differ. For example, in Draftingemosse, the mineral deposition is much larger in the fen part of the system compared to Storemosse. However, the peaks are almost occurring at the same time. Upwards in the sequence, we have a peak around 2,200 years ago, recorded at both sites, 2,200 years ago, and then we have a small peak at Draftingemosse around 700 years ago, which was only recorded at Draftingemosse and not at Storemosse. And from around 400 years ago, dust deposition increased in both sites. Draftingemosse have been analyzed for a longer period than Storemosse, so it's not as clear. And interestingly, we've seen also that peat accumulation rates have increased at both sites consistently. We have parts of the sequences when this is not true, but many similarities between the two sites. And if to put the events at Draftingemosse in a regional perspective, you can see that the general climate in the early part of Draftingemosse was warmer and likely drier at that time, and this had then evolved into cooler and wetter climate. But within these general climate trends, there have of course been episodes of both cooler and wetter episodes. We've also had human activities that increased in the area from around 4,200 years ago. So in conclusion, we have 5 periods of increased mineral input recorded at Draftingemosse, and the peat accumulation rates have increased consistently following the elevated mineral input. And this is also the case mostly, but not always, at Storemosse. And I think that this is interesting to probe further, and the work that Eleanor is doing with the understanding what species is responsible for this would hopefully give us a lot of answers to that. And also, yes, we also had the high peat accumulation event is also recorded here at Raftingemosse. So in general, a lot of similarities between the two sites. And I also want to acknowledge and thank the Bolin Center for funding fieldwork and also conference grants that I've received during these years. And thank you for listening.
Well, thank you very much. Questions?
Thank you for a really super pedagogic talk. That was excellent. I want to ask you a question about the minerals.
Mm-hmm.
When they sit, how long do they last without weathering? I think things like feldspars, 6,000, 7,000 years. Do they? Will they still be there?
Yes, well, there has been some studies that looked into this and found that they are actually surprisingly stored longer in peat bogs than you would expect. Yes. Yeah, and if this is due to that they are coated with organics or why, that's not really clear, but they have been shown to last longer than experimental results would have guessed, shown.
And it was also a bit my question. I mean, to some extent, you capture the dust in the peatland, but dust is also a nutrient for the vegetation. Do you see that the vegetation responds after these dust events?
Yeah, and that's why it's interesting to look at the peat accumulation rates and the mineral dust deposition together. And this is also what's been shown before, that this rapid peat accumulation event there we see not maybe increased dust, but we see increased input of minerals that contain nutrients, like for example potassium or phosphorus. So yes, absolutely. So that's one of the main things.
Okay,
yeah.
so you have different kind of dust, which matters in that case.
Yes,
Okay.
exactly. So not only to have more dust, that will not mean that it will affect the nutrient input, but the composition of the dust.
Okay.
Yeah.
Very interesting.
But also, I mean, regarding the question on the minerals, all minerals will not be preserved in the peat bogs. We are restricted to the ones that we can study, and yeah.
Okay. Yeah. Do we have another?
There's one over there.
Oh, okay.
Yeah. That may seem trivial, but where is the modern peak coming from? Mineral dust input.
Oh yeah.
It all went up.
Yes. Well, most likely this is related to human activities, and like that farming have increased in the area during the last 300 years. And there's also a general trend, which was maybe not so clear in this data, but it's a general trend from around 2,500 years ago of a elevated background dust deposition, which is also likely related to human activities, because forestry, agriculture, all of this will make it possible for more particles to be uplifted and transported to the bogs.
Yeah. Do we have time for another question, or we have to proceed? Okay. One quick question. Okay. Now then, thank you very much.
Oh.
And
thank you.
And now I hand over to a bit more recent time scale again.
I could just add that evolution can be very long timescales. So now it's time for evolution of plant economics in a water-limited environment by Magnus Lind, who's at the Department of Physical Geography.
Okay, thank you. Yeah, so Magnus Lind, I'm doing a postdoc at the Physical Geography Department, and this is work I did with I'm doing with Stefano Manzoni, and it's modeling work. It contains some equations, so if you are very sensitive to that, you can maybe close your eyes. It's so I work mostly on modeling. And I mean, as like a motivation to study evolutionary models, and drought and plant drought is this recent events of drought. For example, here you have in southern Sweden, Skåne. In 2017, 2018, there was a big drought hitting that area, and it was very devastating for the agriculture, which is in the lower left diagonal, but also possibly affected then trees and the upper right diagonal. But it's difficult to know the effects of drought and these climate extreme events. Because it's, for example, with trees, it's occurring over a long time, and maybe they are not affected directly. Maybe it can take several years before they actually die, or so. So to study evolution and these long-term trends is very useful to do modeling. And our main questions is how are plants expected to evolve under drought? And as I mentioned, there is one crucial part of this model is the mortality of trees, and this is so we have birth rate and mortality, which we put in this ecological model, and mortality is unfortunately not so well known. For example, we don't know very much about the thresholds for individual trees, when they're dying or not. And there is also these feedback effects from insect attacks and so. For example, here we have the bark beetle, granbarkborre, which you might know is a very bad bug in Swedish forestry, causing a lot of economical, this is very, very costly. So to build the models, we use one theory that is very useful, which is plant economic spectrum, and it was first, I think it was Reich 2014 who came up with this. It's just trying to put all the plants in a spectrum from slow growing to fast growing, and just with one. And you think so. For example, a cactus is growing very slowly, and it's also have a very slow economy, and also very low transpiration. In comparison, you can think about willow trees and very fast-growing species, which have a high transpiration, has high growth rate, and high mortality. So this is sort of the basic ingredient of the model. Yeah, here comes the equations. Okay, so we had we have an hydrology model. We have a population dynamics model, and these are coupled. So the hydrology model is very simple. It's just we actually have two types, one deterministic and one stochastic. This is the deterministic one. It's actually, so it has the precipitation, constant precipitation, runoff, and then evaporation depending on the amount of soil water content. And we also have the transpiration, which is depending then on the number of plants and on this x. And the x is this plant growth speed trait, which is this economic trait, which I talked about before. We have the population dynamics that has recruitment and mortality. So if you want to construct these evolutionary models, it's very important to have ecology, as I said, and then you need recruitment and the mortality. These are the two basic components. So to study the evolution, we use invasion fitness and a framework called adaptive dynamics. And actually we're looking at how a rare mutant is growing in the environment set by a common resident. So it's a sort of a mathematical trick to get rid of all the complications with the genetics and so. So we just compare a resident and a mutant strategy. And it's a simplified way of looking at evolution. And what we get out of this is three strategies. One, minimizing soil water. And one is the maximizing plant density. And then we have the ESS, which is the evolutionary stable strategy, which is then a strategy that cannot be invaded by other any other strategy. Okay, so the population dynamics, I will not say too much about this, but it's we have, as I said, recruitment and mortality, and the recruitment is just a saturating function of W. Mortality is a decreasing function of W, which is the soil water content. And then we have this optional, we can switch on and off density dependence on the, we have the S and the Q. And by setting S equal to 1, we can have density dependent on the recruitment. And by setting Q equal to 1, we can have density dependent on the mortality. Then also we have this background mortality, which is important, which is the MB. Yeah, and of course there's a lot of functions here, so we simplify them with these power law functions. We have then the coefficients which we fit to data, and the exponents actually we have to guess or estimate. We look at, what we did is just look at, there are a lot of random numbers, and from that we could see that, yeah, actually it matters a lot, but if we can work with a very simple exponents, just setting all the exponents to 1. So that worked fairly well. Okay, so the evolutionary analysis can be very difficult, but in this case it's not so difficult. We have the we use this sort of colorful plots to illustrate the evolution. So we have the resident strategy on the x-axis and the mutant strategy on the y-axis. And by then plotting the invasion fitness, which is the growth rate of the rare mutant in the environment set by the resident. We can produce these plots. We just look at the positive parts. We color the positive parts yellow and the negative parts blue, and we get this PIP, which is the pairwise invasibility plot. And there, this is the, you see the arrows are then, yeah, so by looking at the plot, we can see that the evolution will go towards this point in the, which is marked by the red arrows. So then we can change the environmental variable, and we see then that the ESS is changing with background mortality. And in this case, evolution is always minimizing the soil water content. And this is called a tragedy of the common. It means that the evolution is moving towards a point which is minimizing the resource. And it's very common when you do construct this model that you get. Okay. So one thing we can do with a model is we can get estimates of, or we can make these analytic functions. So here it's showing that the strategy minimizing soil water is saturating, and this is also what the data shows for specific leaf area, which is a good approximation of this plant strategy. And it's saturating for when the annual precipitation is increasing. And one of the most important features of the model was that we got 3 different, when we have density dependent recruitment, so S was 1 and Q was 0. Then we got 3 different strategies. So one was minimizing soil water content. One is the ESS, which is back, and one is maximizing the plant density. And yeah, as you would expect, then when you increase precipitation, you get more fast-growing plants. When you increase the evaporation, you get more slow-growing plants. But for background mortality, it's not. You can get this strange result that actually mortality is, well, first you get more fast-growing plants and then more slow-growing. So this is a little bit reminding of this intermediate disturbance theory, which is saying that plant diversity is maximized at intermediate disturbance. We got a similar result for this stochastic soil water model. And yeah, as a summary, foster plants are expected then to evolve under higher precipitation or lower evaporation, and we could expect both or either faster or slower plants to evolve under a higher background mortality. So thank you very much for your attention.
Thank you. Any questions?
No.
Oh, yeah.
Okay, this may be obvious, but this is not my field at all, but what's a faster plant?
Faster, yeah, that's as I said in the beginning, you can, faster plants they're growing faster, they're also dying faster, and they have a higher transpiration. So it's a sort of energy consuming plant, or energy, I mean, it uses up a lot of resources to grow. Compared to, I mean, a cactus, for example, would be a slow growing plant.
Okay, so
yeah.
So faster refers to the growth rate here.
Yeah, growth rate and also mortality rate and transpiration.
Yeah, this is not my field either, but I've heard that plants growing fast, like trees, they have bigger distances between the yearly rings, and they are more susceptible to fungus and pesticides.
Yeah.
Do you know something about that? We talk about old Norland trees; they are more resistant to this, and better quality of the wood.
Yeah.
Even if it's grow slower and less economic benefit per time unit.
Yeah, I think it's related to the wood density in that case. So slower growing trees have a higher wood density, and also they're then probably more resistant to. So it fits well into this spectrum. Yeah.
Good to know that.
Yeah.
That was partly true.
I'm afraid I think we need to move on. I think there's a lot of poster presentations wanting to come in. They don't know that we're running. We're not running late. We're running on time.
Okay.
So.
Yeah, it's a special pleasure to welcome Karin here. I actually asked her to present here so she can consider her an invited speaker. And the reason for that is she does a paleoclimate reconstruction like many of us, but she looks a bit more careful at actually which kind of species do live back in time and what are the minimum conditions for them, for example, in terms of temperature to exist. And from looking really at kind of biodiversity and thermal limits, she gets actually quite different results than people who just use transfer function based on the modern climate. So I don't want to give you a talk, but just a small story. We don't have much time, but she gave a presentation in Dublin on a big geoscience conference. And she made clear that her talk was over, and there were at least, I think, 7 seconds of silence. So I think that was a very impressive history lecture about paleoclimate. So welcome.
Thank you.
Well, we'll make the best out of it. I think it should be a little bit bigger, because otherwise it gets off. Yeah, it will. Okay, what I would like to present is evidence for less ice and instead prolonged interglacial warmth during the last glacial cycle. I will focus on a site called Sokli. It's situated in a central area of Eurasian glaciation. And it is at Sokli that we have an unusually long, 30-meter thick sediment sequence that reaches back into the penultimate glaciation or marine isotope stick 6 at about 140,000 years ago. These sediments, they have been preserved in the center of a magma intrusion, indicated here, and they occur in a deep depression formed in these deeply weathered carbonatite rocks. So we have this hole in which sediments have been laid down through time, but they have not been eroded away by glaciers. So that's quite unusual for Venus Candia. Well, considering that Sockley is situated in the central area of Eurasian glaciation, one would expect a lot of glacial sediment. Well, the contrary is true. Actually, we find very thick lacustrine and fluvial deposits rich in fossil remains interbedded with tailbeds. So we have now studied this sediment sequence during 15 years. We have attacked layer by layer. We used extensive international collaboration. We analyzed multiple proxies, abiotic proxies, biotic proxies, and we tried to reconstruct climate quantitatively using different methods. And so far we published over 30 papers in peer-reviewed journals. So what I would like to do today is I take you through the entire sequence briefly. I will show some of our results, and then I will discuss the significance of our studies, particular for estimates of global ice volumes. So following in stage 6 glaciation, we find a lake, a deep lake. We reconstruct a vegetation that consisted of boreal forest. And our pollen-based climate reconstruction, pollen, those are microfossils of plants. So the pollen-based climate reconstruction that we have for this 12-meter long lake deposit, it shows that July temperatures exceeded present-day values by about 3 degrees Celsius. And later on we see the same for the winter temperatures. Well, the fossil pollen assemblages that we extract from the sediments, they have close modern analogs, and that is shown here in a short analog distance. And those fossil assemblages nicely plot on calibration samples south and then southwest of Sockley. And this indicates what seems to be an increase in oceanity near the end of stage 5E. Stage 5E is also being referred to as the Eemian interglacial or the last interglacial. I need some water. This was a long time waiting. Okay. And as the lake gets increasingly shallow, we find a fluvial deposit. We find fossil remains also in this upper, more fine-grained part of the deposit. And again, similar during the Eemian. Oh, sorry, I forgot. Yeah, we are here in the upper part. We have the fossil remains. And what we reconstruct based on that is that there was a braided river pattern. We had a steppe tundra vegetation. And again, we have July temperatures which exceeded the present day values. And if you take these 3 lines of evidence together, it would point to a strongly continental climate regime during the cold states MIS 5D. Then during warm states 5C, we most probably had enhanced humidity, because it is at this time that we record a meandering river pattern, because we find here a thick lake deposit that accumulated in an oxbow lake. Well, the pollen, we also study those in these lake deposits, and they show a boreal forest very similar as that we reconstruct for stage 5E. Well, this oxbow lake, the sediment that were accumulated in this oxbow lake, it's here indicated in brown. They are exceptionally rich in macrofossil remains of plants and of animals, and they nicely allow us to reconstruct the infilling of this lake through time, but also it shows a series of so-called plant indicator species, and those are plants that have a very strong relation to mean July temperature. They need a certain mean July minimum in order to flower and reproduce. So here are indicated a few of those values needed by these plants in order to be there at Sockley. And this together again indicates that during 5C, we also had July temperatures that exceeded present day values by about 2 to 3 degrees Celsius. Well, during stage 4, let's see, where are we? During stage 5B, we see it's a cold stage again. We have glaciation of the Sockley Basin, but then the area gets deglaciated again. We see another organic rich lake deposit. It was deposited in a relatively small and shallow lake. And again, the polish allows us to reconstruct a vegetation development that started with a birch pioneer phase. Then we see pine coming in and we go to a pine-dominated coniferous forest. However, this deposit is truncated at the top, so it's possible that we miss the climate optimum or the vegetation optimum for 5A. Also, this lake deposit, it was not that rich in macrofossil, but it has a large variety. And again, we see here plant indicator species indicating July temperatures exceeding present day values by at least 2 to 3 degrees. Then we get the glaciation during stage 4, and we pick up another time that this particular area was deglaciated, and the type of sediment that we now pick up is a glacial lake sediment. Well, these glacial lake sediments, they are very minerogenic, have very little organic matter in them, but we nevertheless try to make a detailed multi-proxy analysis. So we looked for diatoms, for aquatic insects, for macrofossils, for plant zoological taxa. And actually we did this not only for stage 3, when we had this phase of deglaciation. We did that for all the times when this area was deglaciated, so the ice goes away. You get a glacial lake, is water damped by the ice before the whole glacial environment disappears. And although each of these deposits had a very unique signature in this whole stratigraphy, they had many things in common. And one thing that they had in common is again the high July temperatures indicated here by aquatic plants and here by aquatic insects. Well, actually this was the first deposit that we analyzed in this whole sequence, and we were very surprised with those really high summer temperatures. This was a period which previously was thought that Sockley was under the ice. So we already thought, wow, it's quite something that we see that the ice sheet disappeared, but we had not expected those high summer temperatures. And we start to repeat it, and actually it's all very similar as during the last interglacial. So then when you start thinking, it's not surprising at all, because these are periods that we have high summer insolation, here indicated in red, higher than we have at present. So it was always thought in the past everything is cold, but that's not the case, and we can actually explain it because we have high summer insolation. So here is indicated the results from Sockley in a more fancy way. It was for a report for the industry. And so summarizing is, here is indicated how northern Fennoscandia would have looked like during the last so-called interglacial glacial cycle based on earlier studies. And this is how this same area looked like in the same time interval, but based on the research that we are getting from Sockley. Well, one thing that you can see is we take away the ice. We take away the ice and we take away the cold environments. And we not only take the ice away, we put into it really warm conditions. Actually conditions what in geology is called interglacial. Interglacial means at least as warm as today. So based on this, we subdivide the last interglacial glacial cycle into an early interglacial complex that lasted 60,000 years, and then a late glacial part that also lasted 60,000 years. And that's very different from earlier thinking, where the last glacial cycle was thought to have started right after the Eocene interglacial and lasted for about 110,000 years ago. Also, once we are in the glacial cycle, we take the ice away during stage 3. Well, for Sockley, we only have data to fill in this early part of stage 3, but there are studies now coming and being published. One of them is from a site relatively close to Sockley, and it indicates that warm non-glacial conditions persisted in northern Fennoscandia all the way to 35,000 years ago. So it's only like 10,000 years before the start of the LGM, when the whole of Fennoscandia was under the ice. So then what is this based upon? Well, this is based on the long distance correlation of mostly fragmented sediments, and those sediments were very poorly dated. And this picture is very much dictated by the marine record. So it's this record. It's a pity that Raimo is not here. I would have loved to talk to her. So it's this record. It's the benthic global isotope stack, which has been and is still being used by quaternary geologists, glacial modelers, climate modelers, archaeologists as a proxy for global ice volumes. And actually, if you go to the original paper, they clearly state that this curve is influenced by many things. It's water salinity, it is ocean currents, it's water temperature and ice volume. So this curve always have been thought, the last interglacial, it was warm, but then ice starts to accumulate until it culminates into stage 2. And now that we get independent data from the continent, the Sockley site, we can see that this is not a good proxy for global ice formations, and the data needs to be recalibrated. I only talked about Sockley so far, but as you said, at the conference in Ingvar, we had a session, and it's now also from North America that we get robust data, independent data, data that does not depend on correlation with marine data, just data from the continents. Those are long records, they are well-dated sediments, and they also indicate widespread deglaciation. So yeah, to start at the beginning, the Quaternary, it's the last 2.5 million years. Based on marine data, it was always thought to be the ice age with a lot of ice, but now that we get detailed data from the continent for at least the last glacial cycle, we might very much change that picture. We take the ice away and we provide the possibility for much longer and bigger area with substantial warmth, which for this session with plants also creates much more space and time for like plant, animal and human migration. Thank you.
Yeah, thank you very much. I think we don't have time where we can reply to question while the mic is changed. So, yeah.
Thank you for allowing one question. Karin, you reconstructed the vegetation based on temperatures where plants can grow, if I understood it correctly. Yeah, you said there are threshold temperatures, but if I understood it correctly, in the morning, plants can adapt to cold climate and warm. Is that on other orders of time than since the last glacial maximum, or could that affect your thresholds for temperatures? Was it probably less cold?
The deep space talk earlier, or it's
okay.
Yeah. I don't think that plants can adapt to. I think then you are looking at a really long time interval far back in space. But we use what we can do. We use present day climate plant relationships, and we think they are still valid over the time interval that we are working with. I think because we have different, we use different methods, so not just one, to see if they all do similar things like with aquatic insects and plants, and that strengthens our reconstruction.
Okay, sorry for rush on. We had to take more questions afterwards. So the last talk, but not the least, Department from Zoology, Mats Ittonen. Sorry.
Thank you. So I'm Mats. I'm from a PhD student at the Department of Zoology, and one of the best documented effects of climate change in biological systems is that species expand or shift their ranges toward the poles or to higher altitudes. And these species may be, say, threatened species that are forced to shift their ranges, or they may be new agricultural pests that we don't have, for example, in Sweden, that we're afraid will expand in here. So it's really important to understand what their fate will be in the new areas where they're expanding. But even if climate gets warmer and thus more suitable for a species in a new area, there may be other factors than simple climate that these expanding species may need to adapt to. And one complicating factor is seasonality. So even with a fair amount of climate change, butterflies won't really like flying around in the winter in Sweden. So they will have to complete or do all the growing and reproducing in the spring, summer, and autumn. And then they spend the winter in a resting stage that we call diapause. And different species do it in different life stages, but in my study species, it's as a half-grown larva. And depending on species and the length of the season, they may complete one or several generations per year. So, and the number of generations per year is the trait that we call voltinism. And so above we have a univoltine example with one generation, and below a bivoltine example with 2 adult generations per year. And possibility to either, for these half-grown larvae, to either continue development into adults or to enter diapause for winter means that they're making a decision. So these larvae do really have to time their life cycle decision correctly, because if for example these guys don't enter diapause but do like these guys do, they will die because they're trying to become adults in the middle of the winter. So it's really crucial to know what time of the year it is. So how did I do that? Well, the key is day length. So in Stockholm, for example, we have really long days in the summer, and as you currently experience, we have really short days in the winter, and the days get shorter and shorter from summer solstice until winter solstice. And for a caterpillar in July, That means they will, in say early July, they will face really long days, while one in August will face shorter days. And this way they know which decision they should make if they have, whether they have time to grow into adults or not during the same year. So what does this have to do with the range expansions and shifts? Well, day lengths are different at different latitudes. So the day length, that means early July in Stockholm, will mean maybe late July in Umeå. And this is something that the expanding species will have to adapt to when they move northwards. When we are talking about adaptation and range expansions, there are some genetic factors that we need to consider. So here the blue arrow represents an environmental gradient. So we have a pretty bad environment, a good environment for a species in here, and a bad environment to the right. And our species may be really common in their optimal habitat, and then it gets rarer and rarer and may not even be able to reproduce in here or something. And we can identify a core range where there's dense, big populations. There's not a lot of natural selection because they have already adapted over long time periods that they've been there. And there's lots of genetic variation because the population is big, so they're doing really well in every aspect. While in the other end of the spectrum, we have the range margin, where there's strong natural selection because they're not yet adapted to the conditions in there, to, for example, the day length regime. But there's little genetic variation that this natural selection can act upon. And furthermore, there's a strong genetic drift that further decreases that genetic variation. Then there may be gene flow from the core range into the range margin. And because the core range individuals are really well adapted to the core range, they will be badly adapted to the range margin. And this gene flow will make the range margin population less well adapted because these guys are introducing their genetic material into this population. So the big question is, can expanding populations overcome these genetic challenges and actually adapt to the new range? To study this, I have a case of the wall brown butterfly in Sweden, which has pretty recently expanded northwards. And in blue we have the old range, and in brown we have the newer range where it's expanded into. And we can identify a Swedish core range and a range margin. And I'm interested in how the range margin populations have they adapted to the day lengths that they face in this area. An additional interesting factor is that this species has 2 generations per year, mostly in all of Sweden, all of its Swedish range, while many other species shift to univoltinism in their northern range. And these guys don't do it, and it may be a constraining factor that makes them unable to shift even further northwards because the season is so much shorter there. So what we did, we identified 2 populations close to a northern range margin and 2 in the core range, which we're going to compare. We went out in the field, collected butterflies, got famous in Skåne for running around with nets close to old castles. And then we took the offspring of these field collected butterflies, had them in these small cups on their food plants, put them in climate cabinets where we could control temperature and day length. And we had 8 treatments, 2 temperatures and 4 day lengths. And we looked at whether they enter diapause or develop into adults. So we can make a set of predictions of the differences between the populations. So the longest day length where they enter diapause should be longer in the northern populations than in the southern ones, because the same day length will mean later time of year in the northern than in the southern populations. Additionally, we could ask a question about whether they are capable of univoltinism in these populations, and that may be present in both populations, or both in the southern and northern range, but it's more expected in the northern range because the season is shorter there. So to our results. Here we have separately the two temperatures in the graphs, and on the y-axis we have the proportion of individuals that entered diapause. On the x-axis we have the 4 day length treatment, so short days here, long days here, and differently colored lines show the response of the 4 populations, which are color-coded on the map. And then we have the confidence intervals. So what we see is that obviously day length affects whether they enter diapause or not. So in the short days, they all enter diapause. In the long days, almost none of them do. And we have the difference we were looking for. So the 2 northern populations at 16.5 hours, so the next longest day length, half of them entered diapause at that day length, while almost none of the southern individuals did. But in the warmer treatment, we didn't see this pattern as clearly. So there it may be that the threshold is somewhere in between our treatments or something, but at least the temperature also affects the decision that the caterpillars make. To summarize, yes, the northern populations entered diapause at longer day lengths than the southern ones. For univoltinism, we can't really answer that question very well with this data. We would have to sample in a slightly different way. So the big question was, can the expanding populations overcome the genetic challenges and adapt to their new environments? Based on this species and our data, it seems like, yes, they can. So if you're concerned with endangered species, that may be good news. If you're afraid of new pathogens or agricultural pests, then that's bad news. I would like to thank Kalle Gotthard, my main supervisor, as well as Alma, Christer, and Matthew, who have helped or supported me in different stages of the work. And you can also have a look at my poster outside.
Very pedagogical. And you kept time, even short, even though we're running late. So I will allow a few questions.
Yay.
Yep.
Hey, very nice talk. I was just wondering if they can evolve their critical day length, is there any reason to believe that they could not evolve univoltinism?
I'm not sure. They're pretty far from the day lengths that they would have to enter diapause in are pretty long in that case. But I don't think there should be any qualitative thing that would prevent them from doing that. But yeah,
I agree.
Anyone else? I can hear there's a lot of murmuring outside there waiting for come in. But I would like to thank all the speakers. It's been really interesting, a pleasure to have you here.